Good morning, almost afternoon. My name is Robin DeBruni. I am a fish biologist at the USGS Great Lakes Science Center in Ann Arbor. What I'm going to talk to you about today is some larval fish community assessments that we did in the St. Mary's River during the 2022 Lake Huron CSMI. And this was a binational effort. They're sampling from both the USGS as well as Ontario Ministry of Natural Resources. So fish early life history studies in the St. Mary's River, we have pretty limited information. In the 1980s, there was a couple years where there was sampling done in the upper river as well as the mid-river along Sugar Island. And from 2018 to 2021, we did some targeted sampling around the compensating work gates associated with their change in operations. And there's also been some other short-term juvenile and very limited scope larval works that have been done by Lake Superior State, not counting the very extensive juvenile work that's done by the programs that you've already heard about today. But the study objectives for this study was to assess the larval fish communities and the nursery habitat throughout the St. Mary's River. And what I'll show today is our preliminary data. We just finished going through all the samples about 3 weeks ago. So I'll be able to show you our full complement of species, composition, phenology, or when different species are hatching and in what order, as well as some larval densities throughout the river. And so for the river, we had 53 sites. Each one of these dots is our sites. We used paired bongo nets. We sampled only on the surface. They were approximately 5-minute tows, depending on how much debris we got. And for this talk, I've grouped the sites into different sort of sections of the river as we go downstream. And so I've got them into 6 sections to kind of find a way to group and show the data more succinctly. So for our results, we started sampling in the end of April in 2022, and we continued through September 1st. We collected over 40,000 larvae, and we had about 1,700 samples that were collected. We have 22 taxa that we identified, and we got fish from 14 different families. And not surprising, given the habitat complexity of the river when you're going from upstream all the way to downstream, we got fishes from sort of all different community types in the different areas. Just for our community composition, this is based on the densities that we got, not the total numbers of catches. But you can see up here, and we go mostly upstream to downstream as you move from left to right. And here we have our percent composition, and again, that's from the entire sampling period. So we can see upstream we have a whole lot of this dark blue, which is our rainbow smelt. And then all of this sort of brown color, those are our minnows and our shiners. And I talked to the Michigan DNR when they did some surveys later on in the fall, and they said they just saw like thousands and thousands, these swarms of just thousands of cyprinids. So our larval information matches up with that. And it should also be noted that the dominance of the cyprinids is really from only a few tows that were done, where there were, again, 4,000 or 5,000 cyprinid larvae in a single tow. But the other thing other important information is that this light blue color, which is our burbot, we caught burbot throughout the river. This black color, which is our carrigonines, we captured those as well throughout the river. But that mid-river, you can see there's a lot of them that were captured specifically in that section. And the yellow at the top is yellow perch. Oh, and when I talk about perkids, which is the red, and these colors for the species groups will remain consistent in my following slides. That's why I'm trying to point them out quickly. For perkids, that's every perkid except for yellow perch. So I wanted to pull them out because of their importance in the fishery. This is our mean larval densities by those larval sections by week. So we have our mean larval densities over here and our dates along the bottom. Again, you see down here in Muskong Lake, as well as the red, which is that downstream, we have these very high peaks. Those were caused by cyprinids, which we'll see in the following slides. But if you take those out, you can see that all the sections had a peak. It just might have fallen a little bit differently. And again, when we look at the later slides, we'll see that those were caused by different species in those different sections. So for our first two sections, upstream and mid-river, we can see that upstream, this is our, so again, we have larval density along the side, the date along the bottom. I'll show panels, I'll show two panels on a slide, and the y-axes may be different for your comparison, but the colors are the same that were on that composition graph. So for upstream, we can see that we had that peak that we saw up there was caused by rainbow smelt. And for the mid-river, the peaks were caused by lots of carrigines coming up in early May, as well as some cyprinids later on in July and August. Other things to notice is this light color here. Again, that's our burbot. And so we're seeing burbot sort of hatch. They have very extended hatch period that you can't necessarily see because they had the lower densities. And I don't have the growth information, but we were capturing small burbot for 6 to 8 weeks throughout the system. For Lake George and the St. Joseph Channel, we definitely had lower densities, especially for St. Joseph Channel, but we did have, this right here is our yellow perch, and again, these spikes in cyprinids later on in the summer. Now, if we zoom in and sort of allow the cyprinids to go way up, we can see what's happening with the rest of the community. So for Lake George, we're seeing lots of rainbow smelt, and you can see that the peaks for Lake George then kind of match up with what's happening in the St. Joseph Channel. Fish likely drifting down through the system and sticking along the Canadian shoreline. This right here, this perkit is mostly both perkits that we only got to the perkit level of identification, as well as log perch. And we're going to be going through these samples more carefully to look for potential ruff, given what Fish and Wildlife Service has been finding. For Muskong Lake and those downstream sites, you can see we have our yellow perch peak in Muskong Lake, and again those cyprinids later on. And when we zoom in on those, we see we have yellow perch in early May in Muskong, and then we have these multiple other peaks of our other perkids later on in the lake area. And then a lot of those do drift downstream, but we do see that black, which is, again, those carrigines. We're seeing them throughout the river, as well as rainbow smelt, that blue line. So again, compared to the previous studies, this study was more expansive, both spatially and temporally. We did find similar species catches. There was only 5 species reported before, but we have those as well as the phenology that also matched up. They had higher densities in the 1980s for their samples. However, they sampled much closer to shore, and especially for those carriginine samples, if you were here for yesterday's talk where they talked about seining right on the beach, they got much higher densities. That could be one of the reasons for what we found sampling from our small vessel compared to the sampling that was done in Jude et al., where they were really, really close to shore. Something else to note is Fielder in 2000 put out a report suggesting that there was Cisco spawning grounds in what we called the mid-river area, and that's definitely where we saw the highest densities, as well as down in Lake George and in the Muskong Lake areas. So for conclusions, this is the first study to provide that comprehensive measure of larval fish community, as well as nursery habitat assessments in the St. Mary's River. And it can be used as a baseline to evaluate changes either due to habitat modifications, restoration efforts, invasive species, and other things. We can also look at where densities were the highest or where we saw the most diverse community composition, and that's all really the next steps for our analysis now that all the identifications are done. And we found important production areas for sport fish, commercial fish, prey fishes. Our burbot, criganines, and rainbow smelt, we found them really throughout the river, although the timing may have changed depending on which river section we were in. We found, not unsurprising, lots of yellow perch in that Lake George and Monascon Lake area, as well as downstream. And we're also planning to do some genetic confirmations, especially on our criganines. We caught about 1,800 criganines, and we only identified 150 of them as lake whitefish. So that's also what's up for our future. So with that, I'd like to thank our partners and our funding source, Great Lakes Restoration Initiative, Ontario, Ministry of Natural Resources and Forestry, and the USGS. And I'll take any questions.